plan today is to just investigate what is the source, what's the actual source of the EEG and MEG, like what actually physically causes these currents to be able to be recorded at the scalp, and what actually causes these magnetic fields to actually stick out of the head and be able to be recorded with an MEG machine. So, and the problem with understanding this is that there's a number of different levels of organization we have to understand. We have to understand, like, what's going on in the cells, and then what's going on sort of like at a middle scale if we recorded, like with an electrode inside the brain, and then what's going on at the most sort of macro scale when we record, like on the skin, outside of the brain. So, and then eventually next week we'll talk about, how can we localize these signals, both the EEG and the MEG signals. But let's just start off with, with what actually causes them, and it's somewhat subtle. So let's first just consider what's in the cortex, and so. And there's other parts of the brain that you can record evoked potentials from. You can record evoked potentials from the brain stems. Certain very early auditory signals in the brain stem are recordable with evoked potentials. It's possible to theoretically record evoked potentials from the thalamus. But for the most part, when we're talking about scalp evoked potentials, we're mainly talking about recording evoked potentials coming from the cortex and potentially from the cerebellar cortex also. So let's just talk about the neocortex. And so what's in the neocortex? So if you take my other class, which eventually will be posted on YouTube, you can hear a lot more about this. But what are some of the cells you have in there? Well, you've got pyramidal cells, and these are spiny cells. They have little spines on them, and they've got apical dendrites, and they got basal dendrites. So they tend to be bigger excitatory cells, and then they have an axon that comes off, and the axon goes off someplace else, and it might connect up to some other cortical area. But it turns out that most of the connections in the cortex come from cells that are nearby. And pyramidal cells will synapse, they have a whole field of synapses where they synapse on other, I'm drawing like 10,000 synapses there. So they'll have 5,000 synapses on other cells that are in the area. And in addition, they'll have some synapses that go out a long distance. And so these guys are excitatory. And they make up the majority of cells in the cortex. And there's different kinds of pyramidal cells. They're in all different layers. So here's a pyramidal cell sort of in a higher layer, smaller one. And this one, they can actually serve to kind of communicate between layers in the cortex. So this, the axon and this axon might go out. So they can not only sort of communicate nearby them, but they can go to different layers, maybe even a layer that they didn't have a dendrite in. That's a possibility. So you could go and say, make a bunch of synapses up here. There's little stellate versions of these cells that are sort of that don't have a long apical dendrite. So this thing is called the, apical dendrite. This thing, and it's perpendicular to the cortex. So this is the neocortex peel surface, and this is the That's the gray matter, and then here's the white matter. So that's what some of the excitatory cells look like, but there's other inhibitory cells. And the inhibitory cells, one big kind of inhibitory cell is called a basket cell, and every year I always make fun of neuroanatomists. They spend all their time sort of looking through the microscope late at night, and you look at a cell like that, it's just like, does that look like a basket? I don't know. But there's the large inhibitory cells called basket cells, and they tend to inhibit cells that are kind of near. Occasionally, they actually send an axon into the white matter or go sideways through the cortex into another cortical area, but in general, they sort of synapse a little bit more locally than, so that's the red is the axon. So that's, so this is a, pyramidal cell, excitatory pyramidal cell. This guy is, so let's put our inhibitory in here. So this is, they're all in the same place, but I'm just drawing them. So these are, excitatory. So that's an inhibitory cell, which will tend to lower the membrane potential when it synapses on another cell instead of excite it. So that guy is, basket cell. But then there's a bunch of other ones, like one cool cell type, which is present in all different layers. It just looks like sort of a relatively non-distinguished sort of cell body like that, but it's got these axons that come off and form these little, they're called chandelier cells, because they look like, like an old style chandelier, which means candle, sort of like something to hold a bunch of candles. So, and where are those things synapsing? If you blow up a pyramidal cell, there's the very beginning of the axon. So here's the cell body, cut off there. There's a very thin part of the axon right at the beginning before it thickens up and gets myelin on it. And that's called the initial segment. And that's where the spike starts, the action potential. So this thing right here is the initial segment of the pyramidal cell. This is a pyramidal cell. And it turns out, if you actually look at that initial segment, there's a whole bunch of synapses on it. And those synapses are coming from the chandelier cells. So they're synapsing on an axon. So normally, like, an axon comes out and the synapse is made onto a dendrite, but in this case, that's what these little chandeliers are. They're actually just little arrays of inhibitory synapses onto the axon. So it's an axonic cell, sometimes they call that. So that's a chandelier cell. Chandelier. And then there's these tiny little cells that are about as big as the cell body of a pyramidal cell, there are little inhibitory cells called clutch cells, little tiny guys. And then there's all different, there's more cell types than this, but I'm just sort of giving you some of the biggies. And then there's another cell, it's kind of got these somewhat elongated, that's the cell body right there, somewhat elongated dendrites, but then the axon comes out in a big sprig, little vertical column like that. It's got thousands of synapses like that. And this is an example of a cell. This cell uses somatostatin as a transmitter, but this guy's a double bouquet cell. Because there's two bouquets. See, I told you, like the neuroanatomists get going a little crazy. There's two bouquets of axon synapses coming out of the cell, and these guys actually selectively hit inhibitory cells. So they actually inhibit inhibitory cells, which could potentially cause excitation. They probably are partly responsible for columnar excitation in the cortex because they inhibit other inhibitory cells like clutch cells that are up here, and then that disinhibits the area up there, and then you get a little possibility of activity. So that's just a god-awful mess. I mean, look at that. You know, how are you possibly going to understand that? What's going on in there? You know, there's immediate feedback. You're sending things long distances. There's, you know, all different kinds of axons coming in from other places and making synapses in there. You know, and it's sort of changing over time. You know, complete. You know, it just sort of seems like a hopeless case. And so, what can you actually see from this? What would you possibly expect to see from this at a distance? And so to understand that, we first have to sort of understand fundamentally how neurons behave. So let's just take a pyramidal cell. So here's a pyramidal. It's just a cartoon of a pyramidal cell. So the pyramidal cell has a membrane on it, and the membrane has a very high resistance. And so if you make a electrical model of what the pyramidal cell looks like, the pyramidal cell dendrite looks like, you basically have, you can think of it as a bunch of resistors that have relatively low resistance running down the length of the apical dendrite. And then you can model just a one-dimensional model of the membrane as having a very high resistance. So this is a very high resistance. And then there's some capacitance in there too, which we can ignore. And so this is a little chunk of the membrane. It's a model of a chunk of the membrane. And then we just assume that the outside of the cell is all hooked up to itself. And so here's another little chunk of the membrane. And it's got some capacitance also, and another little chunk of the membrane. So this is called a cable model of a neuron. And where did it come from? It came from people trying to figure out, like, is there going to be any current left if we inject current into a telegraph cable that goes across the Atlantic? You know, before you go through the trouble of actually laying the cable, you should try to figure out, like, if I stick something in this end, is anything going to come out, or is it going to all leak out along the way? And so how does this work? Well, this is, you know, resistance, you know, longitudinal, and this guy is, you know, resistance membrane. So, you know, the longitudinal resistance, that's kind of the goo inside the cell. That's pretty conductive, not much resistance. So this is low, and this is high. And so that's why it's a cable. It's a cable because it's like an insulated cable with a small amount of stuff leaking out. And that understanding of how the actual electrical properties of a cell behave is critical for understanding why we end up with an EEG. Now, remember, there's going to be a bunch of other pyramidal cells. Here's another pyramidal cell sitting right next to this one. So basically, how does current flow in this cell? Well, if we've got a channel here, so say there's a channel here, and that channel, say that was a sodium channel. So here's some sodium, and so maybe it was a synapse from another cortical area. That is going to cause the sodium to come into the cell because there's more sodium outside the cell, so it's always trying to get in. So you open up that channel, some sodium goes in, and it's generally going to run through this low longitudinal resistance a long distance. And then, you know, a little bit of it will come out here through that high resistance of the membrane. So a little bit of it will come out, but most of it's going to run down there. And so what what is that going to look like if we record some, if we do an electrical recording, like right in the cortex itself? What it's going to look like is some current disappeared here. So that's what we would call a sink. Some positive current disappeared. So positive current disappeared into the void. Into cell, where we can't get to it. So that's a sink. So we'll just do it for one channel. And then what's going to happen in other parts of the cell? We're going to see current sort of erupting out of the void, and so that would be a source. So a source is positive, current erupting. I like that word, erupting out of the void. And what's the void is the inside of the cell where we can't see it. It's insulated, the insulated inside of the cell. So basically, that's fundamentally what makes it possible to actually see any current at a distance. It has to do with the fact that because of this cable-like property of the dendrites, meaning they've got a conductive inside and then a very high resistance membrane, if you inject current into them, it's going to tend to flow in a radial direction. Now you could have the exact opposite. If you put an excitatory channel up here. So what happened if you put an excitatory channel up here and open that one? Well, then positive current is going to disappear over here, and then you'll see it sort of erupting out of the void, down here. So in that case, that would be a sink because the current's disappearing into the cell, and then this would be a source down here. So that's all source and sink means. It just means, is current sort of like appearing out of nowhere, or is it disappearing into nowhere? But it turns out you need more than just the cables. So there's three critical things that you need in order to be able to see. So here's three conditions to be able to see a signal at a distance. Sig at a distance. So the first one is we need some anisotropic cables. Anisotropic cables. So they have to be, that just means they can't sort of be like a sphere. They got to be sort of elongated so that we can get this current flow to go sort of long distances. But it doesn't help if they're all in every which way, so they have to be aligned. And that would be like, there has to be a whole set of pyramidal cells, that are all, lined up with each other. But even that isn't enough, because if this guy gets stimulated at the top and this guy gets stimulated at the bottom, say like, there's sodium coming into the top of this guy and sodium coming into the bottom of this guy, well, at a distance, those things are going to cancel each other out. And so we also need, coherent laminar. So what is laminar? It generally means like just layer-wise. So we need, we basically have to stimulate, all of these guys down near their cell bodies, and or all of these guys, up near the end of their apical dendrites. So this is, an upper layer, lower layer. So those have to be stimulated at the same time, because any signal that, if we stimulate this one here and this one down here, is going to cancel. And so the bottom line is, and then finally, what about a cell like a basket cell? Like a cell like a basket cell, what does it look like? It looks like it's got something, sticking out this way, something sticking out that way, something sticking out that way. And so if you open up some synapses on, say, the outer part of a basket cell, what's going to happen, you'll get some, currents that are going to sort of flow around like that. And when you look at that from a distance, you're not going to see much. And that's because it's not anisotropic. It's kind of radially symmetric. So any of these more radially symmetric cells, especially smaller ones, is just going to disappear. And so the important point here is that we have to sort of consider that when we record and evoke potential, we're not seeing all of the activity by a long shot. We're not seeing all of the activity. We're only seeing activity in anisotropic cells that are really elongated, basically, and that are aligned with each other. They have to be aligned, otherwise we're not going to see it. And then they have to be coherently stimulated, like excited at the top, or just excited in the lower layer, or excited at the top. So any other activity, of which there's a lot. So cells are stimulated at the top and the bottom at the same time all the time. That happens all the time. We can't see that. We can't see most of the inhibitory cells. So there's another thing to keep in mind. The excitatory cells are like about 80% of the cells in the cortex, and the or 80 or 90%, and the inhibitory cells are like only about 20% of the cells in the cortex. So that's another strike against the inhibitory cells, because there's many less of them, and so you're not going to see as big of an effect. But not only that, they tend to be sort of more isotropic in their dendritic structures, and so that's also going to make you not be able to see the inhibition. So like, you're not going to see anything from chandelier cells, or probably from double bouquet cells, or these other inhibitory cells, or basket cells. So evoked potentials see a limited picture of what's going on. However, it's an actual online processing that's actually occurring, in the cortex that you can directly record. So compared to standard fMRI, which is sort of just recording the exhaust, like the oxygenated exhaust of the oxygen supply of the brain, it's much more direct. You're actually recording the real signals that are some subset of the real signals that are occurring in the cortex. Okay, so any questions about sort of like the fundamental source of cortical evoked potentials? And so what is it? It's basically anisotropic cells that are aligned, that are coherently stimulated in a particular layer. That's the only thing that will make a visible signal. Any questions about that? Okay. Yeah, there's a lot of papers on the structure of the cortex and how it works, and not very well understood. We'll talk later in this lecture about some of the patterns of layers, like where does the input come in to the cortex, and what layers does the output come out of. So this is like at the very micro level of how cells work. And so what I want to do now is talk about microelectrode recording, and then sort of graduate up to recording from the scalp. And for this purpose, we need to talk about sort of microelectrodes, like recording action potentials that occur in cells. But then there's also something called a field potential, which is kind of the sum of all the combined current flow in all the cells in a local area. And then finally, there's an actual scalp potential that's coming through your skin into an electrode that's on your scalp. So starting off at the micro, so here's Here's the micro level, which we already did, but let's just talk about it again from the context of recording with a microelectrode. So if you put a electrode in the cortex, you can record spikes. So what does an electrode look like? Well, it's typically, and back in the day we used to make them, you would get like a fine piece of tungsten wire and then sharpen it by electrolyzing it until it's got a really sharp point, and then coat it with something that's an insulator. So let's coat it with glass. So here's an insulator. So it's only got a little bit of a small little tip. And you know, what does small mean? It means like, say, 15 microns. That's about the size of a cell body. It's like so like 20 microns would be 5 times, would be 1/50th of a millimeter. So it's a small, not a visible exposed tip. And so if you stick that into the cortex, you can record spikes or action potentials. And what do action potentials look like if you do that? Well, they're all upside down. They look like this. There's the spike, and then then there's the recovery. So that's an action potential. Or they call it a spike. And so first of all, why is it upside down? Well, it's upside down because let's put a cell here. So let's put a cell like right about here. So there's the initial segment, and here's the pyramidal cell. So this is our 15 microns. So it's upside down because what's happening is some sodium channels are opening there, and sodium is rushing in. So sodium is rushing in. And so what does that look like to the electrode? It looks like sort of a sink. You see, like, current is leaving the electrode and disappearing into the axon. It's going this way too. It goes both ways. And what's the point of that? Well, the point of that is that somewhere down the axon, say here's where the myelin starts, and way down the axon, there'll be another node of Ranvier where so this part's all sort of insulated with myelin, whose purpose is to reduce capacitance, incidentally, not insulate really. It's mainly affects things by reducing capacitance. But the hope is that enough current will have gone down through the axon so that it will get down into here and cause the sodium channels here to open up and then allow more current to flow in. So here's more sodium channels opening up. But it turns out if you record a lot, I've recorded a lot, usually you would just reverse the spike so it looks like it's supposed to be, like, it's supposed to go up like that and then look something like that. But if you record long enough, sooner or later you're going to actually sort of see a right side up spike. Now, how could you get a right side up spike? You could actually make a really teeny tiny pipette that gets really small, like 1 micron, and put that pipette into the cell. And if you put a pipette like that into the cell and record from that, obviously it's kind of hard to get it in there without damaging the cell. But if you got it in there, then you would see a positive signal when the spike starts. And what's that positive signal coming from? Well, it's coming from the sodium that got let in at the axon initial segment, and also sodium that got let in, from a channel here that eventually goes into the pipette. And so then the spike would be right side up when the cell spikes. And so how could you see a spike that's right side up if you weren't inside the cell? And I know this has occurred. I'll be recording, and so like, well, that's a right side up spike. And how do I know I'm not inside the cell? I just like ding the electrode a little bit, which would tear the cell, and it's still there. And so the reason is that the cable reasoning that I used before. So what happens is sodium rushes into the cell here, but then some of it sort of leaks out up here. And so if you put the electrode, if you happen to put an electrode outside the cell right around here, you'll see this backwash. You'll see the backwash from the cell. You won't see this. So you'll basically see a source from that, as opposed to a sink. This looks like a sink, and that corresponds to that negative going part. So if you ever read neuroscience papers about single unit recording, this is usually what they're talking about. They say single unit because they don't really know what they're recording. You're kind of recording blind. But you put an electrode in, and as you slowly move the electrode, you'll eventually get close to a cell, and you'll start seeing a spike appearing when you get within sort of this range of a cell. So that's the actual sort of signal that's coming out of the cell going to the next cell on the line. So that's, in some respects, that's the real signal that we want to see. But how does that sort of lead to something like an evoked potential? Now, it turns out if you go into the white matter, so this is an important point, if you just take this, say here's the white matter, so here's the gray matter, and here's the white matter. So what's in the white matter? Well, there's a gazillion axons going every which way. Spikes, chaos of spikes, going every which way. What happens if you stick an electrode like this down into the white matter? Complete silence. So when you record spikes, you're actually not recording the spikes going down the axon. What you're recording is the fact that there's kind of a separation between essentially, where the current is disappearing into the void here from the place where it's kind of leaking out. You actually can't see any spikes in the white matter with an electrode tip like a 15 micron electrode tip. And the reason is because what happens is some sodium will rush in here, and that should create a little sink. But the problem is there's some inhibitory channels right there, and then some potassium is going to rush out and cancel the sodium that rushed in. And so if you use a teeny tiny pipette with a 1 micron tip, then you can start to see spikes down in the white matter. But if you use like an electrode with a tip that's about the size of a cell, won't see a thing. You can tell you've gone into the white matter because the signal just disappears. You typically play the signal through an audio. You often play it through an audio monitor so you can hear the individual action potentials as little ticks. And as soon as you hear it, you go into the white matter and nothing, quiet. So that shows that even to record a spike, you're not really recording the spike as it's going down the axon. You're recording something really having to do with the cell body and the fact that the cell body is a cable that's elongated, and so it has a tendency to allow you to separate the source and the sink by enough of a space so that you can sort of actually see them. So that's the micro scale. And it's relevant because that's the real signals that are being transferred between neurons. But what about sort of a medium scale? So it turns out if you record, you can also medium scale. So you can also see something called a local field potential. And so what is the local field potential? So if you record from one of these electrodes here, this is a microelectrode, you're going to see, when you might have sort of been thinking about this, like, what about, okay, this is one cell, but what about other cells? Aren't there other cells there? Yeah, the whole thing is just stuffed with cells, and they're all doing stuff. Synapses are opening on all those different cells, currents flowing every which way. How can you sort of like, see anything coherent? But if you add up all the effects of all the other currents, all the other channels on all the other cells, withdrawing charges from the intracellular fluid or ejecting them out, you get something called a local field potential. And what that local field potential it'll look like sort of a lower frequency change in voltage. So this is, millivolts going that way. And then what you'll see, if you look carefully, there'll be some, spikes on there. So they're upside down spikes, right? So there's an upside down spike, and there's an upside down spike. Depends on how close you are. That spike might be bigger if you're right up on a cell. But you'll see these, you'll see these spikes on top of there. So that's basically the total, total microelectrode signal. Total microelectrode signal. And so what does it have? It has spikes in it, and then it has these larger changes that are sometimes called local field potentials. And so what do you do if you're recording, if you want to record the spikes? Well, you would just filter out the low frequencies. So you run it, you basically just turn the bass down. That's what a high-pass filter means. So, turn the treble up, turn the bass down. And so what happens if you do that? Then you'll see a nice signal like this with just the spikes in it, because all those lower frequency signals will just be filtered out. And then we sort of reverse it so that it looks like it's, like the spikes are right side up. And so, but this is the actual signal that's coming into the electrode. And then vice versa, you could just do a low pass filter if you just want to look at sort of the overall, in that case you would just end up with, this signal with those high frequency spikes filtered out. So that's just, low pass, turn down the treble here, turn up the bass, and then you'll just see the low frequency. So this is total signal. So the evoked potential, the local field evoked potential is present in this electrode, even though people don't often draw it. It's there. It's just got like these very high frequency spikes in it, and then these low frequency changes that are the sum total of all the ion flows and all the surrounding cells, all summed up and totaled. Okay, so that's like a medium scale. So, but we're still not up to a scalp electrode. So how do those local field potentials end up being effectively visible through an electrode stuck to the skin? And before I talk about that, I just wanted to give just a little cartoon of how different cortical areas communicate with each other, because the kind of scale of neural communication that we can detect with a scalp electrode is more or less about at the level of a cortical area or a couple cortical areas. Real cortical areas are pretty small, but we can detect activity in a cluster of cortical areas that's maybe 1 or 2 centimeters across. That's the actual scale that we're capable of detecting. And a lot of cortical areas are in the range of 1 centimeter or 2 centimeters. So if we look at a lower cortical area, and like for example, like V1, primary visual cortex, there's a series of layers in the cortex, which we divide into. So we'll call them 1, 2, 3. Stick 2 and 3 together. 4, 5, and 6. And just a caricature of what these areas are. This layer 1, you can call that the original input. Like if you look in a turtle, this is where all the input. There's still input there in a mammal, but that's the original input. This one is the feed-forward layer, layer 2, 3. That's kind of sending to a higher area. So here's the same thing. Here's a higher. I put higher in parentheses because there's a lot of feedback, and sometimes it's hard to tell who's higher than who. Say for example V2. So lower, higher. So that's going to be another area like that. It's going to be 1 centimeter or 2 across, have, millions of neurons in it. And it's got roughly the same layers in it. So it's got, 1, 2, 3, 4, 5, 6. So now, what are the other layers? So layer 4 is the input layer. Like the current input layer as opposed to the original, archaic input layer. Layer 5 is the motor output layer. And layer 6 is, you can sort of think of it as the feedback layer. Now this isn't exactly correct, but it's sort of correct. And so if you look at an even lower area like the dorsolateral geniculate that's providing input to V1, what does it do? It comes in to layer 4 and synapses on layer 4. And then the input layer will then synapse to these other layers. It will, cells in the input layer will send synapses to get the signal out of the input layer. And then the feed-forward layer is going to go and project to layer 4 of the higher area. The feedback layer, cells in the feedback layer, will go back to the geniculate. Cells in layer 5 are going to go to motor output. And in V1, what is motor output? Well, the superior colliculus moves the eyes. So in V1, the superior colliculus is the motor output. Comes from neurons in layer 5. If you were in motor cortex, well then, the big layer 5 neurons that move your body all around, they're coming out of layer 5 too. And if we go out to this higher area, we can see, it's the same thing that's going to happen over here. They're going to, the signal is going to leak out into the other layers like that. And then by the time it gets down to layer 6, we can see that layer 6 is going to come back and synapse essentially in every layer but layer 4. So skip layer 4 and then synapse on these other ones. So it's feeding back information into the previous one, but it doesn't stick it into its input layer of the previous one. And then if you look at the feed-forward output layer of this higher one, well, that's going to go out and synapse on a higher area. So this sort of gives you some sort of very gross picture of how the cortical processing happens at a more of sort of a middle scale. So these things, these areas are, several, centimeters across. And those are things that could potentially be distinguishable by electrodes on the scalp. But that's still sort of middle level. And so what we want to finally get to is, the macro scale. So the macro scale of a recording and evoked potential. And so what does that look like? Well, now we're going to have some skin. So here's our electrodes stuck to the skin with a little scritchy scratchy, so you get a low resistance connection. So that's the skin here. But then, what's under the skin? Well, there's a little fat under the skin. We'll leave that out. Then there's the skull. Skull is pretty high resistance. There's some debate about exactly what the resistance is. The standard number is supposed to be 80 times as resistant to current flow as the skin. Some people have suggested maybe it's more like 20 or 30, but in any case, it's more resistant to current flow than the skin is. And then below that, we get our I'll draw it bigger than it is so that I can make a nice picture. So here's some cortex below that. So how do we actually get a current that appears through the skin in the scalp? So let's draw one of those cells. So here's a pyramidal cell right here. Bunch of pyramidal cells, and let's say that we strongly stimulated the basal dendrites part. So let's say that this layer of the cortex was strongly stimulated. So what's going to happen? Well, there's going to be, in general, some sort of current, coming out of the top of the cell, because there's a sink down here, and there's a source, erupting out. It's going to go in other directions, of course. Sort of a source kind of erupting out kind of the left side here. But now what happens when it gets to the skull? Well, it's going to say, oh, I don't like that. The skull is high resistance, and so I'm going to flow mostly this way. So the skull is like a higher level cable. So a tiny bit will go through here, little teeny tiny bit will go through here and get to the electrode. But most of it's going to sort of run into the skull and just take the path of least resistance, which is, through the fluid that's inside that high resistance skull. So here's that, high resistance skull. So what does that mean? That means we've got another, so basically we have, a higher level cable. It's not. It's a weird cable. It's a sphere-shaped cable, but it's a cable nevertheless. So it's got low resistance inside of it, but then very high resistance going through the skull. But all our signals are recorded by actually currents that came through the skull. So little tiny bits of current are going to sort of mainly go that way, but just a tiny little bit goes through the skull, and that tiny little bit and then goes through the skin to get to the electrode. So you can see there's another problem, which is what you generally call the forward solution in EEG and MEG, which is if some current flowed somewhere here in the cortex, what would we expect the signal to appear? So it turns out, if current flows anywhere in the cortex, every electrode will be touched. So here's an electrode, further away. There's actually going to be some current coming through that electrode. So it doesn't matter where you are. There's going to be some on the scalp. There's going to be some current tiny currents coming through the skull and getting to the electrode from just activating one spot in the brain. So every electrode will see an effect of this sink and then source, erupting of current from the top of the cell. So that's the bad final news. So you've got this higher level cable, and so we have to make a model of what this does, and so we'll talk about that next week when we talk about the forward solution. And that's a model of how, if you put current someplace, how does it flow through the head and come out through the electrodes, which are what we're recording from. So there's kind of like two levels of cable. The first cable is the one that actually makes the signal visible at all because of these elongated dendrites that are lined up with each other, stimulated in a laminar fashion. But the second cable is when that current, when that cable here generates a signal, it's going to run into this skull cable because the skull is an insulator. So that's basically EEG. So it's measuring currents that come through the skin and through the skull in order to allow them to be recorded from one of these EEG electrodes that were primarily due to these radial flow in different parts of the cortex. So radial meaning like perpendicular to the cortical surface. So this is the peel surface right here. And this is the bottom of the gray matter, and then here's the white matter down here. That's the gray-white matter border. So finally, that just leaves MEG. And MEG is, once we understand this, then MEG is kind of the same thing. So all MEG is the magnetic fields generating by these currents. Now, there's a tiny, tiny, tiny magnetic field generated by these tiny, tiny currents that are going through the skull, not a very big magnetic field. And so we can kind of ignore, so we can't ignore the skull and the skin for doing EEG because all our signal is coming through, and the skin is important too. If there's a bump on the skin, that actually will affect the signal quite a bit. It's a high resistance bump on the skin. But for the purpose of MEG, for the magnetic fields generated by these currents, then we can kind of ignore the currents that are going through the skull. So the MEG forward solution is a little bit easier. And so how does MEG work? So now let's back off to a nice big level. So here's person. So they've got some current that was flowing in that direction. So what is that current? So that's a J. So that's a current flowing in a particular direction in the skull. And what is that going to do? That's going to, if you use the right-hand rule with your thumb being the current, if that's the direction of the current, it's going to be going into the skull there and then coming out of the skull down here. That's essentially doing the same thing as curl. And so if we draw that contour map, what is that going to look like? And what am I drawing in the contour map? What I'm drawing in the contour map is the length of the magnetic field vector. So EEG is a scalar. It's a scalar voltage that's coming out of this electrode here. MEG is a vector. So magnetoencephalography, that's what that means. So this is a vector. It's a vector B, and so that vector that vector is coming out. It's sort of coming out here. Like that. So it's going to be really strong here, a little less strong here, a little less strong here, and then it's going in over here. So here's where it's going in. So that thing. So that thing is the current, like the current that was flowing, like that current. So that's our J. And there'll of course be, in order to be able to see the signal at all, you've got to have a whole zillion other pyramidal cells, and they all have to be doing the same thing. So there's a whole bunch of them in here, and they're all doing the same thing. And that gives you kind of like a coherent, J going up like that. And then that's going to create this magnetic field, but the magnetic field this thing is a B vector. So that's not only has an amount like a voltage, but it's actually a vector quantity. So there'll be a magnetic field coming out of the head that is due to the currents that were flowing in the radial apical dendrites of the pyramidal cells. And like I said, there's a tiny magnetic field coming from the currents going through the skull, but it's so small it's too small, you just basically ignore it. So all you have to do is consider how the currents are flowing, not just in the pyramidal cells, but how they're flowing through the rest of the head. So you have to make a model of how the currents, how these currents flow through the head. So we can make a forward solution for what is the relationship between a current and what is the size and direction of the magnetic field that we would expect from that. And so that's our fundamental sort of two methods of recording, and they're kind of complementary. It's best to do both of them because you can see, like if this current is flowing that direction, what is that current going to look like? Well, there's going to be some current coming out over here, and then some current disappearing over here. So this would be negative over here and positive over here, because some current disappeared here and then came out over here. Whereas the magnetic field is the other way around. It's sort of at right angles to it. Here we have magnetic field sticking out in this part, and then magnetic field pointing back in this upper part. And so it's if you have both kinds of signals, you can do a better job of localizing where the currents, the original currents that were this radial flow in the apical dendrites were coming from. Okay, I think that's the end of that. So that was like micro, sort of middle, and then macro scale of what we're actually recording when we record EEG. So any questions about that? So this is, you can see it's like just paying attention to the exact scale, at which your signal is recorded makes a lot of difference. Like you don't see any spikes. Spikes are long gone in the EEG signal. They're all just kind of averaged out. You don't really see that. You kind of see summed local field potentials. That's essentially what you're seeing in an EEG signal. Summed local field potentials that you would have recorded with an electrode down in the cortex, but now you're just seeing them after they've leaked through the skull. Okay, any questions about EEG and MEG, sort of like where the signal comes from? How do we actually record it? We just record currents coming through the skin, or we record magnetic fields generated by currents that were inside the brain, but then those magnetic fields protrude outside of the brain as a B vector, and we can actually record that B vector. Questions about EEG, so source of EEG, MEG, and sort of at several different scales. Oh, good. Probably not good, but in any case, we're going to charge forward and talk about, next time, talk about the forward solution. So first I'll talk about current source density. So you can think of current source density as the bottom line, like that's actually what happened in the brain. And then we'll talk about trying to localize signals, the forward solution and inverse solution. That'll be next. But first, current source density. And to understand the current source density lecture, it's good to go and look at the grad, div, and curl lecture that I did at 8 o'clock today. Okay.